working from the Institute of Laser, Photonics, and Biophotonics under the supervision of my mentor, Dr. Falarin Oloputo. So today we're gonna cover a few things. First, I'm gonna discuss some background information on silicon quantum dots, followed by how we approached our experiment, then the results, conclusion, future research, and acknowledgement. So what are silicon quantum dots? But before I speak about silicon quantum dots, first I have to define quantum dots, which are semiconductor particles that range from 1 to 10 nanometers in size. So they're very, very small. Silicon quantum dots are also semiconductor particles, but what differentiates them from quantum dots is its core element, which is silicon. And because of its core, because of the silicon within the quantum dots, it displays unique physical properties that have acquired a lot of general interest from researchers everywhere in the fields of chemical engineering and nanotechnology. Reasons why these silicon quantum dots are so interesting is due to its photoluminescence, which is the ability to emit light under photon excitation, and its low toxicity. And the reason why it has low toxicity is because carbon, which is a main element within living organisms, is common, like I said, in living organisms, and silicon is right under it. So it's pretty much the next best thing. And because of these properties, it shows great potential in biological applications such as cellular imaging and cancer diagnostics. And this is an example of the silicon quantum dots under UV light. So these are examples of biological applications. This is a picture of an in vivo picture of a mouse. Here, so here are the quantum dots. It has orange luminescence in a mouse tail. These are the cells within the tail. And then this is a regular image of a cell with the quantum dots inside under luminescence. So what's the problem with silicon quantum dots? Although they have a lot of great potential, they do have their limitations, which is typically after synthesis of these nanoparticles, they have a hydrophobic core. And the fact that it's hydrophobic is a big problem, especially for in vivo studies, because living organisms are composed of water as well. So, and hydrophobic means that it's not friendly to water. So that's a problem right there. And also, when used in vivo studies, since the cells are so deep inside the body, it could be the nanoparticles could be brighter. So the issue that my project focused on was the water dispersibility issue. And one hypothetical way that I addressed the water dispersibility issue was to encapsulate the quantum dots with compounds that have hydrophilic groups, thus creating a shell around the quantum dots, which would render them more water dispersible and biocompatible. Okay. So one compound that we use that fits the criteria is called chitosan. Chitosan is a naturally based polymer that exhibits biocompatible, biodegradable, and hydrophilic properties due to its amino acid group and hydroxyl group on its surface. The thing about chitosan also that is very beneficial is that it is used commercially in like fat pills where when you ingest it, it absorbs fat within the body, so it's very biocompatible. Also, this is the image of chitosan. As you can see, this is the hydroxyl group, OH, and this is the amine group, NH2. And this is a schematic I drew, basically showing what we're trying to do. So, I'm having trouble with this. Oh. so the white and red dots are basically the chitosan, and the orange particles in the middle are the silicon quantum dots. So these polymers will encapsulate the dots and therefore make them more water soluble, and it will increase its biocompatibility for biological applications. So our approach was just to give a general overview. First, make the nanoparticles, then reduce its size, then modify its surface, and then make it biocompatible. So first, in order to make how we made the nanoparticles was through laser pyrolysis, which is the end result would be these nanoparticles. So we used an aerosol laser reactor, and you probably saw this in Tao's presentation, but this is the reaction chamber where the reaction, first, in order to make the nanoparticles, you have to decompose silane gas, which is SiH4, and then the end result is Si plus H2. So this is where the reaction takes place, and this is the end result. Which are the powders, and it doesn't have this luminescence yet. Followed by etching, which basically is in this step the nanoparticles gain its luminescence, and also we reduce the size of the nanoparticles. The main point in reducing the size is to get a specific color. So each color corresponds to size. So red is the biggest, and then followed by orange, then yellow, then green, and so forth.